Taping? Okay.
All right, folks. A couple of things we need to do. The big handout I gave that I didn't staple together, it's all about DNA and some of the some fine points about DNA that some of it you know, some of it you don't know in there. And I do want to go through that, but and we would have if we had had a full class period today. But we do have a lab coming up on Thursday, and it's important that I stick to Thursday lab schedule because that's when Stephanie Wonder shows up, and she can't make it on Tuesday, so we have to make sure we do our labs on Thursday so she can help out on that. That's kind of the point that we're coming. So I do want to get a lab going Thursday, so that means we need to get prepared for that. So let's talk about some other stuff. You can put the DNA stuff off to the side for now. In fact, we won't get to that today. Two topics that we want to talk about here today, and then we probably won't take the whole time to talk about those, and then give you some time to work on an assignment that you need done. So, people watching the video, you got an assignment to do for next time. All right. One topic is restriction enzymes. And we went over this in 10th grade, so I think you pretty much knew all of this at one time. I'm not sure just how much Mr. Vanoverlo covered it. I know I covered it. And you probably are getting the same handouts that you got in 10th grade. I don't remember exactly which ones I gave you then. Who was in Mr. Vanoverlo's biology class there? Do restriction enzymes? Do you remember them?
A little bit, yeah.
Okay. All right. And then the second thing we want to talk about today is Gel Electrophoresis. And I know that both classes did that. In fact, I think you did a lab with some Gel Electrophoresis. And I think you did one with the restriction enzymes also. So some of this is review, but these are two tools that are used probably every day by many, if not most, molecular biologists. They go to work, and they know they're going to run a gel, and they're going to do a digest, a restriction enzyme digest. It's just tools that you use all the time. These have just been incredibly important tools, incredibly important. Well, this was a discovery. This was not a discovery, a what? Innovation. Innovation, yeah, we'll call it an innovation. That's good. All right. Let's talk about restriction enzymes first. A little bit of history of it. Scientists, I don't remember the years, I should look up some of these years, but I think we're talking the 1970s probably. Scientists were wondering, why don't bacteria die from viral attacks? Viruses do attack bacteria. And when a virus attacks us or a bacteria, what it does is it injects its DNA into a cell. So a virus really isn't a living cell. It's just a piece of DNA or RNA wrapped with a protein. And when a virus lands on you, for example, if it lands on your skin, it can't do much. It can't get through your skin. But if it gets into your eyes or into your nose or into your lungs or into your blood if you have a cut, then what it's going to do is it's going to inject its DNA into one of your cells. And its DNA says one thing, that is, make more of me. And so essentially what happens is it takes over your cells and converts them from doing whatever they're supposed to do to doing just one thing, and that is making more viruses. So your cells just start pumping out viruses. And pretty soon, the cytoplasm gets filled with viruses, and the cell bursts. And it spews out millions of more viruses, which then infect the neighboring cells, and that's an infection, a viral infection. We, of course, have an immune system that helps us to fight against that. We have antibodies, like we talked about in the last lab, that fight against those things. And usually we win, unless you're getting one of these very nasty tropical and African ones like Ebola and SARS, which isn't tropical, but things like that. But when we get common cold, the flu, usually our immune systems gear up in time and we get the upper hand. But scientists are wondering, well, what keeps bacteria don't have an immune system? How do they stop viral infections? And so they were studying this, and they found out that bacteria have things called restriction enzymes. And these restriction enzymes chop up viral DNA. And so here's a bacteria, no nucleus, of course. They're prokaryotes. And here comes this virus that lands on it. This is the virus. And it injects its DNA into, make it double-stranded. It injects its viral DNA into the bacteria, and sitting here in the cytoplasm of the bacteria are these restriction enzymes. Restriction endonucleases is what they're generally called, right? And these restriction enzymes chop up this foreign DNA and make it ineffective. Well, researchers realized that, hey, this is a tool that we can use, because one of the problems that was plaguing researchers at this time was how to cut DNA in specific places. You know, you can't see DNA. Maybe you can see a big wad of it. I think we all extracted it in 10th grade, didn't we? And this looks like this blob of snot. I'm sure, yeah, I know we did. I think we did. No, Mr. Yeah, I think we did. Anyway, so you get this blob of snot-looking things. Yeah, that's the DNA. But you can't see the bases, the A's, G's, C's, and T's, and you can't stretch it out and measure it, and cut it here and there. The best that scientists could do, if they wanted a piece of DNA, is they could just shake it violently, and it would fragment, and it would just fragment randomly. And so they never knew what size pieces they were going to get. And so that was always a problem. You come on a bus?
No.
Okay.
It took me an hour to get here.
An hour?
Yeah.
Traffic or?
Yeah, I got on the highway. It was a dead stop.
Come on. How many times did I tell you, don't take the highway in the winter? It's a terrible place to terrible thing to take, because all it takes is one accident and you are sitting still. At least when you're on the side roads, you can get off and go some other road. Okay, so scientists wanted some way to cut DNA in the same place every time. And what was special about these restriction enzymes is not only that they cut DNA, but where they cut DNA. And they found that a restriction enzyme always cuts DNA in the same place every time. But we got to get you some handouts here. So was there an accident or just slow?
There were probably 5 accidents.
These you can just put away for another time. And we just started, by the way. I just put off till. All right, and so to make it a little more clear, take out one of your handouts there. So as this comes up, take this one out. All right, and so what researchers found was not only do restriction enzymes chop up DNA, but they always chop it in the same place. And there are dozens, today maybe there's hundreds, of different restriction enzymes that have been discovered, and each one cuts in its own unique place. What we mean by that is this. Here's one called EcoR1. This is a common one. We will be using this one on Thursday, EcoR1. And EcoR1 looks for a GAATTC. It has to find a GAATTC DNA sequence. It recognizes that. Not only that, most of these restriction enzymes recognize palindromes, which means that the antisense strand is GAATTC. So you read this one that way and you read the same one that way. Not all DNA is like that. Palindromes aren't that common. So EcoR1 looks for palindrome, and that's the palindrome. And when it recognizes that DNA sequence, it will cut. But it doesn't cut straight through, and this also made things very, very useful. But rather, it cuts between the G and the A, so it cuts right there. And so that breaks the backbone. We've got this sugar phosphate backbone up here that we usually leave off when we draw DNA. But it cuts through there, and then it breaks the hydrogen bonds this way and then goes down through the G and A again. So it gives you that kind of a cut. And we'll see why that's really important, very useful. And so now scientists had a tool. They had a scissors. They had a DNA scissors that will always cut at the same place. So if you have a length of DNA, let me just draw it as one line. So there's our double-stranded DNA, and there's what we call a restriction site, that is a GAATTC right there, then you can always cut that DNA right there. So if you have 1 million copies of this piece of DNA, you can always cut in that spot on all 1 million of them. Before this, if you had 1 million copies of this, all you could do is shake them, and some of them might break there, but some might break here and here and here and here, and they could break all kinds of places. Now, scientists had a way to cut one in the same place every time. So why was that important? Well, because this sequence, for example, will likely show up more than one time. In fact, if we took all of our whole genome, all 3 billion base pairs that we have in every cell, and you cut it with EcoR1, there would be thousands of restriction sites in it. But if you take just one piece of one chromosome, and there's a gene in there that you want to cut out, all you have to do, let's say, for example, that this is a piece of DNA, and here is the gene that you are interested in. All you have to do is find a restriction enzyme that has a recognition site, a restriction site there, and there, and you can cut that gene out every time. And so you can target where you want to go. Yes, some questions?
How do they know what kind of restriction enzyme to use? Like, how do they know what? Like, if they want to sequence, how do they know?
You can go, because so much has been sequenced, so much DNA has been sequenced today, that you can go online and read the DNA sequence of almost any chromosome of a human and of many, many other animals and plants as well. And so you just have to look at it and you say, okay, I want to cut there and I want to cut there. In fact, you can start engineering them, and you can start putting pieces of DNA there that have a restriction site and say, I want to cut a little closer, so I'm going to stick a restriction site in there, and then I can always cut that. So those are engineered genes that you can work with them. Yeah.
Once you separate that gene, how do you make sure that you get it and not the other two pieces? Is it like obvious? When you
yeah, you're right. Good. It's gel electrophoresis, and so that's the natural follow-in to go to gel electrophoresis where you find that piece. All right. So we have all these different. you've got HinD3. I think we're going to be using HinD3 as well on Thursday. Then there's BAM and TAC and ALU and HAG and BAL. And there are dozens of these things. And you see that they all have different recognition sequences. So if you want to specifically find a piece of DNA, all you have to do is pick the right restriction enzyme, and you can cut out your piece of DNA. All right. One more thing about this, and that is how do you name these things? How do we get these weird names, BAM and ECO and all that kind of stuff? And I do want you to know this. What you do is you get the name from the species of bacteria that it was discovered in. And so the first letter of the restriction enzyme, in BAM H1 for example, comes from bacillus. It was found in a bacillus bacteria. And then the species name of that bacteria was Electrophoresis. And you take the first two letters of the species name. So the first letter of the genus, the first two letters of the species, and it gives you BAM. Bacillus Electrophoresis. Then, sometimes there's a letter behind them, like here, this one is ECO. By the way, this one came from E. coli. Escherichia is what E stands for. Coli, everybody's heard of E. coli. And then you have oftentimes a letter behind it. With bacteria, you don't name them like animals and plants. Oftentimes they have a strain letter behind them. It's kind of a subcategory within the species, and they're called strains. So this was strain R. In BAM H1, it was strain H. Right, so you just have to kind of know what strain it was. And then finally, this is not a letter, this is a Roman numeral, a number. It designates which one this was that was found, because there can be more than one restriction enzyme found in each of these guys. So this was the first one that was found in this genus and species, that strain. Look down here at HinD3. Haemophilus, that's the H. Influenzae, that's the IN. Strain D. And that tells you how to pronounce these two. Not Hind, but HinD. And it was the third one found in that particular genus, species, and strain. So that explains where we get these names from. All right, and this explains it. For those of you who might be taking the exam, almost sure there'll be questions about this, so hang on to these sheets. All right, you remember this from 10th grade? Got it. Good. I don't want to beat a dead horse, so let's move on. So then the question is, well, how do you separate these? How do you know you're getting that one? And then we do gel electrophoresis. So let's just briefly go through gel electrophoresis, or at least quickly go through it, because I think you probably remember it too. So gel electrophoresis is a way to separate DNA fragments of different sizes. When we're talking about DNA, you can separate all kinds of things. You can separate proteins, you can separate dyes, but we're separating DNA. And so you have this gel, and you have these wells in them, and this is going to be part of the lab on Thursday. I'm going to have you pour your own gel and do the whole process. And so if you have a test tube here, and you have a mixture of fragment sizes. You have some DNA fragments that are long, some that are very, very short, and some that are halfway in between. So let's say we have 3 different fragment sizes. So we've chopped this DNA up into 3 different fragment sizes. And we know that the target gene is on the, let's say it's on the medium-sized one. And so we pour all of those, we take our pipetters and we fill our wells with those, and we turn the electricity on, and you know that the DNA starts moving. And it moves in a straight line. That's kind of a cool thing. The electrical current going through here just pulls that along in a straight line. If it starts straying all over the place, you'd have all kinds of problems. Anyway, so you leave it run for about an hour, and after an hour, all of the small ones will have gone the same distance. Now, do the small ones go further or?
Further.
Yeah, because they're fast. They get through the little beads. All right, so after about an hour, your small ones are all going to be down there. And your medium-sized ones are going to be someplace. It isn't necessarily a linear relationship, so we'll just put those there. And then your longest ones, depending on how long they are, the really long ones are going to stay way back here. And again, it depends on what kind of sizes you have. If you have one that's 20,000 base pairs long, they're not going to go very far. The ones that are 100 base pairs long are going to go a long ways. And so you now know that this is the DNA that you were targeting. And you can actually tell how long these fragments are by running a ladder. We usually run a ladder. We always run a ladder. And a ladder is simply a mixture of fragments of known sizes. So we run a ladder and we get that. So I call it a ladder. It looks like one. And if we know what fragment sizes were in our ladder mixture, then we can actually put, let's say this is 16,000 base pairs, and this one we know is 15,400 base pairs, and so on. And we can label all of these, and in that way then we can come pretty close to determining what the length of these fragments was. Just a little bit of a reminder again, some students are sometimes confused as to why the lane, the band I should say, is always the same width as the well. And the reason is, I think I explained this once in 10th grade, but if this is our well, let's just make it big, and we get a fragment that's as wide as it, that doesn't mean the DNA was that wide. That doesn't mean the DNA pieces were that long. It doesn't mean that at all. Even your longest fragments of DNA, the very longest ones, are minuscule compared to the size of the well. So even your longest ones are very, very small compared to the well. But you have so many of them that they go from one end of the well to the other. So these guys will all move this far, these guys will move that far, and these guys will move this far. And there's so many of them that you get a solid line of them. But if you could actually look in that line, you would find that there are millions of little fragments in there. Okay? All right, good. All right, now let's get to what we're going to do. And I think I have another handout for that. I don't know where it is. Yeah, in fact, we're going to do that. That's what this lab is about. We're going to get the DNA back out of the gel and do something else with it. All right, so here's what we're going to do. We're going to take a piece of DNA. It's lambda phage DNA. Phages are bacterial phages. bacteriophage. Anybody remember what a bacteriophage is? 10th grade? You remember the name. It's a virus that attacks bacteria. All right, so these viruses that attack bacteria have a special name. They're called bacteriophages. And scientists have taken the DNA out of bacteriophage lambda. It's just the name of that particular virus, lambda. And they do a lot of research with it. It's just a convenient one to use. I believe it's 48,502 base pairs long. You have a handout on that. And what we are going to do is we're going to take some of that bacteriophage lambda and put it in a test tube. And then we are going to restrict it, that is, cut it with, and I can't remember which one we're going to do first. If we're going to cut it with eco. Okay, so we're going to do what's called a restriction digest. A restriction digest. Then we are going to take that out of there and put it into a gel. And we only need one well. And so we'll take the results of our restriction digest and we will put it in here. And we will expect a certain number of fragments. It all depends on how many restriction sites there are in lambda phage DNA for eco. And that's going to be part of your assignment to figure that out. Anyway, we will get more than one band. All right. Now, we're going to take one of these bands that has a restriction site for HinD. So then we're going to take this gel and we are going to extract the DNA out of the gel. Take that DNA out. Put it into a test tube. Does this show up on there where I am?
Yeah.
Okay. Put it in. And we are then going to restrict it with HinD3. And then we will run another gel with it, and we will make a prediction first before that's going to be your assignment to predict what you're going to see in your gel. But what you should see is, well, how many bands should you see in this new gel? Well, it depends on what? Excellent. It depends on how many restriction sites there are for HinD on this fragment. So that's another thing you're going to have to look out for this assignment is how many restriction sites are there in there, and then you will predict what your gel will look like. All right? Think you get it? All right. So here's your assignment. Oh, take this restriction map out that you have. This is a restriction map for Lambda DNA, and it's a restriction map. Now, first of all, up on the top, you see the entire genome for lambda phage DNA. It looks like it's 50,000 base pairs long. Below that, figure 5 is a restriction map of BAM H1. So this, if you cut it with BAM, this is what it will look like. And you see that you have a restriction site at 5,505 base pairs, then at base pair 22,346, then at base pair 27,472, and so on. And so how many restriction sites are there for BAM in lambda phage DNA? 5. Looks like there's 5, correct. And then don't forget that you have your end pieces then that also will give you fragments. Then on the top of the restriction map, you see that you have some numbers there. That indicates how long each fragment is. So you could then take this information and you could draw a gel, and you could put bands in the proper spot, right? And they don't line up. Your bands and your gel aren't going to line up from top to bottom, left to right, are they? You have to pick out which ones are the right size. Okay, good. Here's your assignment. I want you to look up online the restriction sites for Eco R1 and Hindi 3. Eco R1 and Hindi 3. What's that? For phage DNA, correct. For lambda phage DNA. And you're probably going to have to spell that out when you do a search lambda. And if you hit the right site, you'll find both of them on the same site. In fact, you might even find, if you go to images, you might even find a map drawn for you. And I want you then to draw neatly with a ruler or graph paper 2 restriction maps.
No ruler?
What's that?
No ruler?
No ruler?
No ladder.
No ladder. You don't need a ladder.
Ruler.
Right. Use a ruler, not a ladder. You can use a ladder for a straight edge, but it's a little clumsy. All right. So what I want you to do then is draw 1 restriction map for Eco R1, and right underneath it, the same size so that everything lines up, a restriction map for Hindi 3. And draw it just like this handout, where you have the restriction sites listed on the bottom and the fragment sizes on the top. All right, then one more thing. Then I want you to take the largest fragment of Eco R1 that gets cut by a Hindi. Okay, so find whatever is your largest fragment. Wherever you see an overlap between that largest fragment and a Hindi restriction site, I want you to calculate what fragment sizes would you have if you restricted that largest section here with Hindi. So let's just suppose, for example, that your largest section is right there, and you find that there is one Hindi site in it. Then you have to say, okay, this one was so many base pairs long, and then if I subtract that out and that out, if I took this section and hit it with Hindi, I would end up with 2 fragments such and such size. Right? And that's going to be the goal of our lab. We're going to first digest it with Eco, run a gel, cut out the largest band, which, how will we know which one is the largest? It'll be the closest to the wells. We will elute the DNA from the gel. We will run another digest with Hindi and run another gel, and hopefully our predicted results will be right on the money. They probably won't, but they should be. If things go wrong. What's that? Yeah, right. Actually, I'll give you a hint. There is one that it's only got one Hindi site, so we should only end up with 2 fragments. It keeps it a little simple. The problem with this is that when your fragments get really small, like in the 100 base pair range, they're really hard to show up on a gel because there's hardly any DNA there in the dye. You have to dye these things. There's no DNA for the dye to stick to because they're so short. The big long fragments, they get all kinds of dye on them. They glow like crazy when you put UV light on them. But the little short ones, you can hardly see them. They're so faint because there's hardly any DNA there to begin with. So that sometimes is the problem with this lab, but I'm changing it a little bit this year, and hopefully we'll get enough DNA there to actually show up. All right, you got it? Okay, and that is due Wednesday, because we're going to do this lab on Wednesday. So all you people out there watching Thursday, all you people watching over there in TV land, you've got an assignment. Yeah.
Is Thursday the day the band has
Yes, so some people are just going to have to miss it. I don't know what to do about these things anymore. I think there's 2 people in the band. Or is there more? Liz probably is too. How many here are in the band? 3? All right. I don't know. You guys are just, I don't know, just out of luck. I don't know what to do about these kinds of things. You're going to have to come in at another time to do this lab, because I don't want you to miss it.
Part of it before this. We don't have to do it.
Till when? You know what? You might be able to do most of it because this is a 2-day lab, because we do the restriction enzyme, and that takes a while for it to work. And so basically what we're going to do is make a gel and do a restriction enzyme, and then next Tuesday we'll elute it. So you can probably get the whole thing done. Good. Yeah. All right. You got the last part of this. You can go to the library and start looking up these. I suspect in 15 minutes we could probably find these restriction maps.